everybody. Welcome to Chapter 11, Part 2. All right, so we have talked a lot about what's going on at the cellular level with muscle contraction. All right, so we're going to take sort of a larger or more universal, I guess, view of contraction. So first we have to define two terms with this. Okay, we're talking about whole muscle contraction. So we need to define two terms. The first one is load. The second term is tension. So what does load mean? Load is simply referring to the weight of an object and or the weight of the bone that's being moved by the muscle. Okay, so simply it's just the weight of whatever is being moved. The second term is tension. So tension refers to the force that is generated by the muscle in order to move the load. Okay. Now, in order for something to be moved, tension has to exceed or overcome the load. And that makes sense. And in order to move a certain weight, there must be enough force generated and exerted on that weight in order to move the weight. Okay, so the philosophy here is tension must overcome load. All right. Now, for the force and duration of contraction is going to vary. You actually had a question on your last lab quiz related to this. So the force and the duration and the amount of time that a contraction takes place is going to be very largely dependent on the stimulation that's coming from a neuron. All right. And so the motor neuron will give different frequencies. It will give different intensities to the muscle fiber. And again, the whole muscle in response will vary in its degree of force of contraction and how long that contraction is taking place. Okay, so main idea is it's mainly going to be dependent on stimulation and input from the motor neuron. Now, contraction, we have talked about how contraction is or can be defined by a shortening of the muscle. That's what we've said. And yes, that's true. Okay? For the most part, you know, when we're dealing with contraction, we're dealing with a shortening of the individual sarcomeres, which collectively, all the sarcomeres in one cell and all the thousands and thousands of sarcomeres in all of the muscle cells, they're going to shorten all simultaneously, all right, and this will result in movement. Okay? So you should know there are two types of contraction. Now, the type we have really been talking about is called isotonic contraction. This is when we do have a shortening of the muscle, and this happens because the tension has overcome the load. Okay? Now, when we say tension, we're talking about the force generated by the cross bridges. So when the myosin head of the thick filament joins with the thin filament, this creates a force. This creates a tension in the muscle. And as long as that tension is enough, as long as we have enough tension, we're going to be able to move or lift a load, right? And this is associated with muscle shortening. Now, there's another kind of contraction called isometric. Isometric contraction is when we do not have a shortening of the muscle. Okay? We do not have a shortening of the muscle. And the reason we do not have a shortening of the muscle is because there is not enough tension to overcome the load. Okay? We don't have enough force generated by those cross bridges to move a load. Okay? So because of that, we will not have shortening of the muscle. So iso, remember that the prefix iso means same okay, or equal, same or equal. So isometric, metric means length. Okay, so in isometric, we are not having a change in the length of the muscle. Okay? Length stays the same. So because we're not having a change in the length of the muscle, right, because not enough tension is being generated at those cross bridges, we're not going to be able to lift the load. Okay? So you should have an idea of those basic definitions here. Okay, like what's the definition of tension and load? And this concept that tension has to exceed load, you know, and what are the basic definitions of isometric and isotonic contraction? Now, I want to clarify. So here's another term. It's called muscle twitch. And I want to clarify to you all that twitch, okay, the muscle twitch, in this case, is not referring to something pathological. It is not referring to something going wrong. It is not a muscle spasm. All right. So let's make that really clear. A muscle spasm is not a muscle twitch. We are going to talk about spasms and how those are caused. All right. We'll talk about two different types of muscle spasms. Understand that when we say, now muscle twitch actually comes from, you know, like muscle physiologists who really specialize in muscle came up with this term. And, you know, the muscle twitch is simply referring to one contraction. That's all it is. It's a simple contraction that comes from the response of a muscle cell to the action potential of the opposing motor neuron. Okay, so understand a muscle twitch in in normal physiological terms is not referring to anything pathological or abnormal. Okay, it's just referring to one contraction. Okay. Now this is recorded on what's called a myogram, and we'll see a picture of that. There will be 3 phases to a muscle twitch or muscle contraction. 3 phases. You have a latent period, period of contraction, period of relaxation. This was also a question on your quiz that you had in lab. What's the latent period? Well, during the latent period, no activity is really seen in muscle yet. This is representing the activity of the neuron at this point, the excitation part, and how we have this activity occurring at the neuromuscular junction, right? We have ACH being released across the synaptic gap, okay? And then we have our ACH bind to the ACH receptors on the muscle, and we're going to have all this sodium influx into the muscle cell, okay? The latent period, okay? We're not really seeing any tension developing in the muscle yet because the myogram is measuring really tension, okay? So in the myogram, we have this latent period where we don't see any tension development as of yet. It's like everything that's happening at the neuromuscular junction just prior to the next phase, which is cross-bridge formation. So this is considered the period of contraction, okay? So the second phase of a muscle twitch on the myogram is called the period of contraction. And this is when we are having tension development. We are having the development of force because our cross bridges have been formed, because our myosin heads are moving the thin filament to the M line. We're having a shortening of the muscle. Okay, this is the period of contraction. We are seeing that our tension will overcome the load. The third phase is the period of relaxation. Now, remember that relaxation is just as important, and there's just as much going on in the relaxation period as the contraction period. Okay, the relaxation period is actually a very active phase. What are we having during the period of relaxation? We're having calcium being pumped back into the SR. Okay, remember we have these calcium pumps on the sarcoplasmic reticulum that are going to be pumping calcium from the sarcoplasm back into the SR. Now, the period of relaxation is actually going to take longer than the period of contraction. Okay, contraction is really quick. Relaxation takes longer, and there's a few reasons for that. Okay, there are a few reasons. The reason this relaxation period is longer is, well, it takes longer for a few things to happen. It takes longer to get all this calcium out of the sarcoplasm. Okay, that takes a little bit longer to do. Also, what else do we have to cause relaxation? Well, we have to have cross-bridge detachment. Remember when we talked about the cross-bridge cycle? And you have to have another molecule of ATP bind to the myosin head to cause that myosin head to detach from the thin filament. And so that itself takes a little bit of time. Okay? So, these things cause this period of relaxation to last a little longer. Okay, so again, the muscle will contract faster than it relaxes. So here's a picture of a myogram and these three phases. Right? So remember, latent period. Now look what we're measuring here. We're looking at muscle tension. Okay? The myogram measures the amount of force being generated. Okay, it's muscle tension. So the latent period, we don't see any tension developing yet in the muscle. Okay? This is just representing activity at the neuromuscular junction. Okay? But once we get all that sodium influx into the muscle cell, and once that causes the voltage-gated calcium channels on the SR to open, and you have all this calcium efflux from the SR, right? And then we get all that calcium to go and bind to troponin, causing troponin to release its hold on tropomyosin and reveal those binding sites for the myosin head on the thin filament. And then we have cross-bridge attachment. So once those cross bridges are formed, then we start to see this increase in tension. And this is represented as the contraction phase. Okay? And the tension will continue to increase. It will continue to increase until the load has been moved. Once the job has been done, once the load has been moved, we no longer need to do this, right? The goal has been achieved. Once the tension has overcome the load, we can now then be signaled to do the relaxation phase. Okay, so here's your relaxation phase. And what is this representing? It is representing calcium slowly being pumped back into the SR, right? And then we're going to have all of our little ATP molecules bind to the myosin head, causing myosin head to detach from the thin filament. And so remember, that relaxation phase is going to take a little bit longer because of that. Those things take just a little bit longer than this excitation part. Here's just another picture of it, all right, a normal myogram. I'm sorry, well, it's a muscle twitch, right, on a myogram. So just another way to look at it, okay? Same thing. Now remember, muscles will vary in their response to what the neuron is telling it to do, okay? So muscle responses can be graded, right? And again, this is going to be based on the intensity and the frequency of stimulation coming from the motor neuron or motor neurons. All right, so responses of the muscle are going to be graded in two ways. They're going to be graded based on frequency of stimulation. Okay? So how often is that neuron firing on that muscle cell? How many times? Okay, so that's referring to frequency of stimulation. Second way that muscle responses are going to be graded or vary is based on strength of stimulation. Okay, strength. So how intense are the signals coming from the nervous system? How intense are those signals? All right, here again is a single twitch. Okay, we have our stimulus. Now, when we say stimulus, we're talking about the motor neuron stimulating muscle cell. Right? And again, we're going to see this little latent period here, and then our period of contraction. up to the point where tension does exceed load. Once that happens, we will go through the relaxation phase. So again, expanding here on how muscle responses vary. We can summarize this by describing it in two ways. We can call it temporal summation or spatial summation. So basically, when we say summation, what does that mean? It means sum, like you're adding something. Like, what's the sum of 2 plus 2, right? It's 4. So we're summing something up. We're adding stimuli together. Okay, so that's what we mean by summation. And there are two types of ways that we're going to add up the stimulation given by a neuron. First, we can do what's called temporal summation. We will add up these stimulations from the neuron based on temporal summation, which is also called wave summation. So temporal really refers to time. So temporal summation means this is a way to sum up the number of stimuli over time. Okay, so temporal summation, what you're seeing is an increase in the number of stimuli from a neuron in a specific amount of time. Okay, so in temporal summation, we're having an increased number of stimuli in a specific period of time. Okay, so this is usually going to be like quick, rapid succession. Now, what happens in temporal summation, or what can happen, if we get too many stimuli, if we get too many in a short period of time, what's going to happen is that the muscle does not have enough time to relax. Okay, so this is referring to a type of, or can refer to a type of muscle spasm. Now, when we say spasm, the word we really want to use is tetanus. Okay? Tetanus means spasm. That's really the formal word. You should definitely know. Okay, tetanus is what a lot of people call spasm, muscle spasm. So what happens here is that a motor neuron is giving a lot of action potentials in a short period of time to a muscle fiber or fibers. And because of all these increased numbers of stimulations, the muscle fibers do not have enough time to complete the relaxation phase. Remember that the relaxation phase is a little longer than the contraction phase. And in order to have normal muscle contractions or regular muscle contractions, you have to be able to complete this relaxation phase to its end. All right, you must be able to complete that. If the cell is not able to complete that relaxation phase, because there are an increased number of stimuli in a short period of time going to that muscle, then you will have tetanus. Okay? So remember, a further increase in stimulus frequency, okay? Frequency refers to number, so the increased frequency of stimulations from that motor neuron means that muscle cell does not have enough time to relax in between contractions or in between stimulations, and so this will cause tetanus. If this continues to happen, eventually the muscle is going to fatigue, and it will have a decreased response to these increased numbers of stimulations, and basically the tetanus is just gonna stop. Okay, so this is like something that we all experience. This is just kind of random. Now, there can be a lot of different causes to this kind of tetanus, a lot of different causes, right? And I'm going to detail out actually 2 different kinds of tetanus, okay, 2 different kinds. It will be important that you're able to distinguish between the 2 different kinds of tetanus. So what's another way or another type of summation? Another type of summation is based not on the number of stimuli coming from a neuron. It's based on how many muscle fibers are being stimulated in a certain area. Okay, so if you have an increased number of muscle fibers over a space, over an area, that's what spatial means here, is over a certain space, over a certain area. Okay, so again, think of temporal wave summation like, let's say, for example, you have one muscle cell and you're having one motor neuron give a lot of action potentials to that one muscle cell. That's temporal summation, right? We're having an increased number of action potentials on that one muscle fiber in a short period of time. Spatial summation is when, let's say, you have one motor neuron that's innervating several muscle fibers, a group of muscle fibers. Again, it's when you have a motor neuron stimulating a larger number of muscle fibers over a certain area. So a larger part of that whole muscle is being stimulated. All right. Now, when that happens, it causes that overall muscle to have a greater strength. Okay. So let's detail out these two types of tetanus. Remember that tetanus is going to result from temporal summation. All right. Now, here's what you're looking at. Let's say, now, first of all, what's being said down here? Unfused or incomplete tetanus. Okay, so this is your first kind of tetanus. There are two types. The first kind is called unfused tetanus, or you could call it incomplete tetanus. Now, what's happening here? Look at the little red arrows. These are representing action potentials. These are representing stimulation coming from a neuron. Okay, remember what our normal myogram looks like. You're going to have a latent period, you're going to have a period of contraction, and then you're going to have a relaxation period. But notice that in this myogram, we do not have a full relaxation period here. We only have a partial relaxation. The reason we only have a partial relaxation period is because, look here, we're having a second stimulation from that neuron coming to that muscle cell. So this muscle cell is not permitted to have a full relaxation period because it's getting stimulated again. All right, so once again, boom, up here, another contraction. Whoa, look, we have another stimulation. So again, we don't have a full relaxation period. Now, if you have this increase in the number of stimulations over a short period of time, you're not going to get full relaxation. Instead, you're going to get a tetanus, okay? What you think of as a spasm. We've all had that happen. Okay. Now, the reason this is called unfused tetanus is because look at this line here. The line is not all straight. And this is another reason why this is called wave summation. Wave summation, okay? This is not a straight line, okay? So it's considered unfused. This line is not fused. It's not all one line, so that's called unfused. Okay, so now what are some causes to this? Okay, again, this is something that we all experience, and a lot of the times, for most of us, most of the time, it's random. Okay, it's random. Other causes to this are going to be, let's say, side effects from certain medications. All right, if someone is taking a medication that where its mechanism of action is associated with, let's say, manipulation of ion channels. All right, what if this drug is going to, let's say, block potassium channels? A drug that blocks potassium channels, well, what's happening during relaxation? Potassium efflux. If you're blocking those potassium channels, remember that relaxation is marked by potassium efflux. Potassium is leaving the cell, remember? Remember that. Okay, so that's just an example. If someone is taking, for example, a potassium channel blocker, they would be more prone to having this type of tetanus because the potassium, you're not having as much potassium efflux, so therefore you're not getting full relaxation. Okay, so this is an example. There are lots of reasons lots of things that can cause tetanus, this kind of tetanus. Caffeine, okay, taking let's say you had a whole bunch of coffee, all right. Stimulants, all right, that have to do with activating sodium channels. Well, you're going to continue to have excitation to the cell, right? Okay, so again, there are lots of different causes to this tetanus, and a lot of the times it really is just random. Okay, a motor neuron just starts firing, and you notice that it goes away. It's annoying, but it goes away because eventually the muscle cells are going to fatigue. They're going to kind of like stop responding to that. Okay, so first type of tetanus is this one. It's unfused incomplete tetanus. Here's the second type. Okay, so the second type of tetanus is called fused tetanus or complete tetanus. It's called fused because look here at our line of tension. It is fused. It's all one line. Now, you see fused or complete tetanus when there are so many stimulations that there is no relaxation period at all. Okay, so fused or complete tetanus means you're having a very high number of stimulations coming from that neuron so that muscle cell is not having any time to do the relaxation period. Now this we've all experienced as well. It's a little more rare. It's much more rare than the unfused tetanus. This is what a lot of people call Charlie horse. All right, this is like when you're getting so many stimulations that the muscle feels like it just locks up. It just locks. All right, and maybe that part of your body, maybe it's the foot. A lot of people get this in the foot, in the toes, and this is what you're going to experience here. It's just a prolonged contraction, and that feels like your muscle is just locked up. All right, so do know the difference here between a fused and an unfused tetanus in terms of temporal summation. What's this? Just showing you the relationship between the strength of a stimulus and, let me go backwards. This is representing spatial summation. Okay, so in spatial summation, remember what you're doing, and this middle picture here sort of illustrates it. So in spatial summation, it's not that you're having an increased number of action potentials going to one cell. It's that you're having an increased number of muscle fibers over a certain space being stimulated at the same time. So you can imagine if you're having more and more muscle cells being stimulated over a certain space in that muscle, that's going to increase the strength of that overall muscle contraction. So again, this is just another picture, okay, of what I've already described, okay, the relationship between the frequency of stimulus and tension. Okay, so this here myogram on the left is just showing like a low stimulus frequency. All right, in this case, we're having full contraction, we're having full relaxation, and we have enough time that muscle cell is totally back to the latent period here. And then we have other stimulation, contraction, full relaxation, we have time to relax. This is a low stimulus frequency situation. Here we're showing temporal summation and incomplete tetanus. So remember, temporal summation, look here at the bottom, we're having an increased number of stimulations. We don't have enough time for this muscle cell to fully relax. Okay, so we're going to get a tetanus. We're going to get an unfused tetanus. And then, of course, over here is the fused tetanus. And look down here, we're having an even greater number of stimuli going to this muscle cell, and there will be no relaxation period possible in that case. Muscle tone, just basic definition on this. This is something that's unconscious. You don't realize that you don't control this. This is something done by spinal cord reflexes. A reflex is unconscious. You do not have voluntary control over your reflexes, all right? So it's a spinal reflex that involves a slightly contracted state of all muscles. And why do we have this? Really, to keep us stable, to stay standing up, or to maintain your position, right? So that's really all we need. Now, of course, our muscles are going to provide input about position this is coming from stretch receptors in muscles. Okay, back to isotonic contractions and isometric contractions. Remember, isotonic contraction means that you are having a change in the length of the muscle. All right? But we're not having a change. You may not have a change in tension. Okay? You may or may not, all right? But this will make more sense. So isotonic contractions, there are going to be two types. Okay? There are two types of isotonic contraction. You have concentric and eccentric. Concentric isotonic contractions are the contractions we really have been talking about. This is when you do have a shortening of the muscle, okay? And it's going to develop tension, and it's going to maintain that tension. It's going to maintain the amount of tension needed to do some sort of work. This is why it says up here with no change in tension. Understand, I don't want you to get confused. Understand that there is going to be an increase in tension initially. There has to be a rise in tension if we're going to overcome the load, correct? But what happens is that tension continues to increase until it reaches the point where it doesn't need to increase anymore in order to move that load. You're only going to develop the right amount of tension, the amount of tension that's needed to do the job. Once you have reached that amount of tension, then you're going to maintain that tension. At that point, there will not be a change in tension, right? Because you've reached the amount of tension that's needed to move the load. Okay, so understand, what's the real difference between concentric and eccentric contraction? Well, concentric is when you have a shortening of the muscle. Okay? Eccentric is when you have, instead of a shortening, you're actually going to have a lengthening of the muscle, a lengthening. Okay, we haven't really talked about this kind of contraction, okay, but understand, what is isotonic contraction? It means you are having a change in the length. So for concentric, we're having a shortening. For eccentric, we're having a lengthening. All right, and this is for specific types of movement. And a great example of this is, let's say you're holding a book, okay, and let's say you're flexed at the elbow, all right? You're holding that book in the palm of your hand and you're flexed at the elbow. And let's say you want to lay that book down on a table that's beneath your hand. And what are you going to do? You're going to end up extending at the elbow to place that book down on the table, right? So you can practice this, all right? And you will notice and feel that the biceps brachii, instead of shortening, in this case, it is lengthening, okay? But it is still considered a type of contraction where we are going to have a rise in tension. And then once we get that rise in tension, we will maintain the level of tension that is required to place that book down. Okay, so two types of isotonic contraction, concentric and eccentric. Here are some illustrations of this. The first one is the concentric type. So with this, so we're showing a muscle, right? And there is a load here. right? 3 kilograms, a load. Now, what's going to happen with concentric contraction is that we see a shortening of the muscle, right? This muscle will develop enough tension to the point that's needed to lift this load, and you see that this results in a shortening of the muscle. Okay, what about isometric contractions? Remember that isometric means that we're not having a change in the length of the muscle. And this is because the load is too much. Okay? We're not able to develop enough tension to move the load. All right, because not enough tension is developed, you get neither a shortening nor a leaning of the muscle. Now understand that tension is being generated, okay? Because the myosin heads are forming cross bridges with the thin filament, but not enough tension is going to be developed to cause any change in the length of the muscle to lead to a moving of the load. Okay, so in isometric contraction, there is no change in the length of the muscle, but we do have tension generated. Now, you see this kind of contraction in maintaining posture. And we have something called antagonism in muscles. And, this is a whole story here that we could get into, but we're not going to. So, but in general, so let's just say, for example, let's take the rectus femoris, for example. The rectus femoris on the anterior aspect of the leg, right? Let's say the rectus femoris is contracting. That would be an agonistic muscle action, agonistic. For every agonistic muscle action, you have an antagonistic muscle action. So what this means is, again, using the example of the rectus femoris, let's say the rectus femoris is contracting. At the same time that that muscle is contracting, there is going to be an opposing muscle that will yield to the action of the rectus femoris. Okay? Antagonistic muscle in that example would be the biceps femoris. Okay, so just in general here, okay? Let's say rectus femoris is contracting. At that same time, the biceps femoris, which is on the posterior part of the leg, is going to yield. It will be inhibited. It will be serving as the antagonist. It will be inhibited while the rectus femoris is contracting. So basically, when one muscle is acting, there is going to be an opposing muscle that has an opposing action that will be inhibited while that other muscle is permitted to contract. This is something that's associated with stabilizing joints as well. Okay, so this is the kind of contraction we would see in that case for antagonistic muscles. All right, what's going on with, just as this is not related to, well, yeah, it is. Okay. Sorry. So go back to isotonic contractions. What we're having, remember, we're having a shortening of the muscle. Okay, actin filaments are being moved to the M line, right? Because, that's going to lead to a change in the length of the muscle if we're shortening. Now, isometric, we're not having a change in the length, right? So what's happening is cross bridges are generating force, but the actin filaments are not being moved. Okay? So we still have tension, but we have no change in the length of the sarcomere. So basically, in these isometric contractions, myosin heads are sort of like spinning on their wheels. All right, they're like spinning around in that same actin binding site, and this develops the tension. But again, no movement. Not enough tension will be generated to move the load. And so isometric contraction, right? We have a heavier weight, 6 kilograms. This muscle will generate force, but it will not be enough force to actually move this load. So again, we're not going to lift the load. There will be no change in the length of the muscle. Just an example of this, right? Isometric contraction here. Okay, this person is not having a change in the length of the muscle. But as she's holding this weight, see how she's just holding it? Let's say she's not moving, okay? What if it's too heavy? She can't pick it up She can't bring it upwards, let's say. Well, she'll generate force because she's holding it, all right? But there will be no change in the length because she cannot overcome this weight. Tension will not be enough to overcome it. Let's say she can lift it. Let's say she is able to generate enough force to lift that load. And look, the muscle shortens. This is considered isotonic concentric contraction. Let's go the other way. This is kind of like when I gave the example of laying the book down on the table. Let's say she has this weight here and she wants to lay it down. She wants to lay it down. This is eccentric contraction. She will have a lengthening of the muscle, okay? And that would be eccentric contraction. All right, let's switch gears here and talk about ATP. All right. So you know the importance of ATP in muscle contraction. What are all the reasons that we need ATP? So this is a reminder of all of the reasons that we need ATP for contraction, okay, for an entire contraction cycle. For one muscle twitch, what do we need ATP for? Well, we need ATP hydrolysis on the myosin head to cause the myosin head to move upwards so that it can attach to the thin filament. That's one reason. Another reason, we need another ATP molecule come along and attach to that myosin head to cause that cross bridge to detach. What's another reason we need ATP? Remember our ATP-dependent calcium pump on the SR. Okay? That's during relaxation. Right? So we need ATP to assist with the relaxation phase. Remember, also, we had something called the sodium-potassium pump. The sodium-potassium pump is going to help maintain the resting state of the cell. I mentioned that back in part 1 of this chapter. We're going to be revisiting the physiology of the sodium-potassium pump again in the next chapter. Okay, but for now, just keep in mind that the sodium-potassium pump, okay, is going to be associated with maintaining the resting state of the muscle cell. And that sodium-potassium pump is dependent on ATP. Okay, so again, you should know all the reasons that we need ATP for muscle contraction. Not just the contraction period, but for the relaxation period as well. So as you can imagine, we're having all of this go on. You're going to need a lot of ATP in your muscle cells because the ATP does get depleted pretty quickly. So what we're going to need to be able to do is regenerate that ATP pretty quickly. So the next series of slides are all about how we're going to regenerate that ATP. First thing to keep in mind is that the ability to regenerate ATP is dependent on oxygen levels. Okay? It's dependent on oxygen levels as well as glucose. Okay? We're going to detail that out. Okay. So there are 3 main ways, and this is really, high yield information. You certainly will have 2 to 3 at least questions about your understanding of these 3 different ways that we're going to regenerate ATP for our muscle cells. Okay, so we're going to go through each process. The first one is just called direct phosphorylation of ADP. Remember, what does phosphorylation mean? It means to add a phosphate. Okay, so whatever ADP you have there, we can regenerate that back into ATP by adding a phosphate to it. Now, there's a certain substance here that's going to be responsible for adding this phosphate back to ADP to form ATP. It's called creatine phosphate. Okay, there's a substance in your muscle cells called creatine phosphate, and creatine will donate its phosphate to ADP to reform ATP. Okay. Now I'm going to talk about each one of these processes individually. Okay, so I'll get back to I will get back to these other two. Okay, but let's just deal with one at a time. Okay, so let's talk about this direct phosphorylation by creatine phosphate. Okay, so ADP again will be regenerated into ATP when creatine phosphate donates its phosphate to ADP. This is also called the phosphagen system. Now, a few things that you want to understand about each one of these three ways that we reform ATP. What you want to know is how many ATP are made by each process. Definitely that will be part of your question, right? You'll need to know how many ATP results from each one of these three processes. Okay, so now creatine phosphate, how does it donate phosphate to ADP? It does it with the assistance of this enzyme called creatine kinase. Remember we talked about phosphorylation means you're adding a phosphate, and that is going to be performed or carried out by a specific enzyme called a kinase. There's another process called dephosphorylation, which means you're removing a phosphate from something, and that is going to be performed by an enzyme called a phosphatase. Okay, so in this case, we're dealing with phosphorylation. We're going to have phosphate. So we have an enzyme called a creatine kinase. It is going to transfer the phosphate from creatine phosphate over to ADP. Okay, so you should know that basic process. So creatine phosphate plus ADP is going to lead to this. This should be a forward arrow here. I'm not sure why that didn't come out. Okay, but creatine phosphate, and then you have ADP around. Well, creatine kinase here is going to take off this phosphate from creatine and add it to ADP, forming ATP. So this is a picture of this, okay? So you should study these pictures because what you want to know is how this works. You want to know how much ATP is going to result from this. So per every one creatine phosphate molecule, you will have one ATP formed. Another piece of information that you definitely want to know about each one of these three processes for ATP regeneration is oxygen required? With direct phosphorylation of ADP by creatine phosphate, oxygen is not required. Oxygen is not required. Okay, so this is a really quick way for you to regenerate ATP, but remember, it's not going to make very much. Okay, it's only going to make one ATP. That's not much. Okay, so that is the first pathway. Okay, the second and the third pathway. First of all, let me mention what aerobic respiration is. Aerobic means oxygen requiring. Oxygen requiring. Now, respiration here does not refer to breathing like in the lungs. Okay, this is something else. Cellular respiration is the process by which we use glucose to form ATP. Cellular respiration. Now, if any of you have taken chemistry classes, especially biochemistry, you've learned about this, right? Cellular respiration is the process by which Glucose is going to be broken down, and we're going to get ATP from this breakdown of glucose. Now, there are two types of this cellular respiration that can occur in cells. One is aerobic respiration, meaning that this process requires oxygen. Or you can have anaerobic. Anaerobic respiration is referring to a way that we can make ATP without the presence of oxygen. Anaerobic means without oxygen. Okay. So I talked about anaerobic. I sort of put this so that anaerobic is next, so let's do it that way. All right. So in the anaerobic pathway, we're going to have something called glycolysis. You also have glycolysis in aerobic. What is glycolysis? Lysis, remember, means to break down or to break something apart. So glycolysis means the breaking down of glucose. Okay, so glycolysis is a normal cellular process that's happening all the time in all of our cells. We are breaking down glucose all the time. Okay. Now, in the case of anaerobic glycolysis, we're doing this breakdown of glucose in the absence of oxygen. Okay, so what this allows us to do is to still make ATP, right, when there's no oxygen. Now, in the anaerobic pathway, this is going to happen like later on in the period of time that you're exercising. So this is when, this is, I don't know how long, it depends on the individual, but I don't know, maybe 30 minutes or so into like continual exercise. If you're just continuing exercising and you're not drinking water, okay, you're not you're not replenishing enough oxygen to your cells, okay, so you would end up doing anaerobic glycolysis. So you'll be producing ATP because there's a demand, okay, but you don't have oxygen. Now, what this will lead to is the formation of something known as lactic acid. Okay? And we'll detail this out in a picture. Okay? Now, you get just a little bit of ATP with this. All right, so let's look at that anaerobic pathway. I know I'm sort of jumping ahead and going back, but right. So anaerobic pathway, right? We're going to have glycolysis happening. We're having the breakdown of glucose in the absence of oxygen. And this is going to lead to lactic acid formation. Okay, and let me actually jump to the picture on this because it's, I think, going to make a little more sense. Okay, all right. So here is the picture. We have glucose. Okay, glucose coming from our diet, or let's say we're breaking down glycogen storage inside of our muscle cells. In any event, we have glucose present. And glucose is going to undergo breakdown. Okay. Now, what's going to happen is at the end of glycolysis, the end product, and you should know this, the end product of glycolysis is a molecule called pyruvic acid. Very important. Okay, so again, the end product of the process of glycolysis is we get this final breakdown product called pyruvic acid. Now, glycolysis is quite a process. There are many, many, many steps to it. And throughout the different stages or steps of glycolysis, you will get the production of other molecules. So, for example, throughout the process of glycolysis, you will get the production of 2 ATP molecules altogether. Okay? So, you will get 2 ATP per glucose molecule in the process of anaerobic glycolysis. Okay? So, you should definitely know this is not in the presence of oxygen. We're doing this. No oxygen use because there's no oxygen available. Look here. These are oxygens. There's no oxygen available. Okay? But the glycolysis process itself, all by itself, will, without oxygen, still produce 2 ATP without oxygen. Okay, so that's fine. Now, why am I mentioning this pyruvic acid down here? So here's the deal. Remember the organelle that does ATP synthesis? This is way back from chapter 3. What organelle do ATP synthesis? That is called the mitochondria. You remember the mitochondria? Mitochondria is responsible for ATP synthesis. So here's the deal. As the end product of glycolysis, pyruvic acid must enter the mitochondria in order to start the process of ATP synthesis in the mitochondria. I'll say that again. Pyruvic acid, as the end product of glycolysis, must be able to get into the mitochondria. Okay, pyruvic acid has to enter the mitochondria in order for ATP synthesis in the mitochondria to begin. Okay, pyruvic acid starts that process of ATP synthesis in the mitochondria. Now, here's the deal. In order for pyruvic acid to get into the mitochondria, oxygen must be present. Pyruvic acid will bond with an oxygen-containing complex, which transports pyruvic acid into the mitochondria. Okay, so following that story, if you don't have oxygen, you can't get pyruvic acid into the mitochondria to start ATP synthesis. So pyruvic acid can't get into the mitochondria. So what happens to it then? We have to do something with it. So what will happen is the pyruvic acid will be converted into something else that's useful. Pyruvic acid will be converted in the absence of oxygen. It will be converted into something called lactic acid. Now, lactic acid will then be released into the bloodstream, and it will be sent to places like the liver. Now, the liver has a special capability. The liver is able to do something called gluconeogenesis, which means, and you don't really need to know that term. I'm not going to ask you about it. But in other words, the lactic acid goes to the liver because lactic acid will participate in actually reforming glucose. So then the liver can do glycolysis again, okay? But first we're going to make glucose from lactic acid. It's a crazy little process. It's a crazy little process. So the liver is going to reconstruct this whole situation, okay, and make and eventually end up getting energy from this lactic acid, okay, by way of starting this process again. All right. Now again, remember this is in the absence of oxygen. So again, when someone is exercising for a long period of time, and let's say they're not taking any breaks, and let's say they're not hydrating, they're not drinking water. If they're not taking breaks and they're not having water, they're not replenishing their muscle cells with enough oxygen. So this is what will result. You will have lactic acid production, and this will be released into the blood. And now this is the feeling, a burning sensation. You will feel it. It's a burning, the lactic acid going through the bloodstream. This is an acid, all right? It's going to cause a burning sensation. All right, so what you need to know here is this process, including everything I said about mitochondria and pyruvic acid, it's in the absence of oxygen, and you're going to get 2 ATP. Now let me go back a few slides to the beginning of this process to make sure I did mention everything. Okay, so anaerobic glycolysis. Okay. We can get the formation of 2 ATPs through this process and it does not require oxygen. Remember that glycolysis is the process of breaking down glucose, and one of the end products of glycolysis is the formation of this pyruvic acid. So we need to get that pyruvic acid into the mitochondria, but it can only get into the mitochondria if oxygen is present. Low oxygen levels prevent pyruvic acid from getting into the mitochondria to do ATP synthesis. Now, a lot of times ATP synthesis is also, it is part of the respiration process. Okay. Remember, pyruvic acid will enter the mitochondria to start the aerobic respiration phase. But again, if you're having high intensity activity, and you're not replenishing yourself with water and everything, you won't have enough oxygen to do normal glycolysis. Another way that this is going to be caused, again, with either long endurance or like endurance training, if you're exercising for long periods, but also if you're doing high intensity exercise, what can happen is that the muscles, as they sort of swell out, are going to place pressure on blood vessels, right? So this can diminish the delivery of oxygen to your muscle cells, right? So that's another way that we're going to see this, is not just with long periods of exercise, but with high intensity exercise as well. Okay, so remember, in this situation, pyruvic acid is converted to lactic acid, and we will send that lactic acid through the bloodstream. It will be sent to the liver, but it's also going to be sent to other really important organs, like the heart. Okay, vital organs like the heart, we will see this lactic acid go there and help provide energy for the contraction of the heart muscle, okay? Because we have to keep our heart going, right? Kidneys, all right, liver. Remember that these organs can use lactic acid for energy, and then the liver is really good at something, and that is actually called gluconeogenesis, where it uses pyruvic acid it uses lactic acid to recreate or remake glucose, as well as pyruvic acid. Okay, so aerobic respiration. I know I'm going back. I just want to make sure I cover everything. So I'm going back to the initial slide here. I talked about the three. Okay, so aerobic respiration, then, this is the type where this is ideal. This is the best kind, okay? This is the best, most preferable way to make ATP. And this is going to be done in the presence of oxygen, okay? So if you have enough oxygen, you're going to be able to make a lot more ATP. This reiterates the importance of drinking water. Again, I know I've said it before. Everybody is always emphasizing, oh, enough water. Well, this is why. This is why, right? Because we need that oxygen in water. Okay? We need that oxygen that you find in water to help make ATP molecule. Because what's the oxygen making up? These phosphates. Remember, the phosphate has a lot of oxygen attached to it. So that's why we need all that oxygen, and therefore water. Okay, so aerobic respiration, we're going to make a lot more ATP this way. It's the best way. Of course, you're not going to get generation of lactic acid, so you will not get that burning sensation. And again, we're talking about a continual supply of oxygen here. Okay. Right. So aerobic respiration, this is really the main type. This is going to be responsible for making 95% of the ATP that you make at rest and light to moderate exercise, light to moderate. All right, so what happens is, glucose is going to be broken down into several things. You don't need to know all the things that it's going to be broken down into, except for pyruvic acid and ATP. We're going to get a lot of ATP from aerobic glycolysis, and you should know how many. Per one glucose molecule, for every glucose, you get 32 ATP. That's a lot. So here's a picture of the ideal situation. This is the aerobic pathway. We have glucose. It'll be broken down into pyruvic acid. Look here. We may not have a whole lot of oxygen, but looks like we've got some oxygen here that we can use as part of our oxygen-containing complex that will transport pyruvic acid into the mitochondria. And then it's inside the mitochondria that we're really going to do a lot of this cellular respiration, all right? And you can know this other stuff. Don't worry about this stuff. If you want to know, you can know. But main idea here is how much ATP is made. We're going to get 32 ATP per every one glucose molecule that is broken down. Oxygen is required for this process. So this picture is just showing you like what we're going to be using, which one of these processes we're going to be using given time. Okay, so in the beginning, you just start out exercising. You just start. You're doing aerobic respiration. Okay, remember that you have myoglobin. Myoglobin in your muscle cells stores oxygen. Okay, so with aerobic respiration, it's very easy. You don't need to replenish your oxygen levels at this point. You're just starting. Okay, it says here 10 seconds. Now this is all relative. It depends on what kind of exercise is this person really doing. I mean, are they running very lightly? Are they walking briskly? They're probably not going to go into the phosphagen system at that point. If they're running very intensely, or exercising very intensely, in a short period of time, then yeah, we're going to go ahead and kick in that phosphagen system where we have creatine phosphate participating in producing 1 ATP. Okay, and then again, it's just duration of exercise. Okay, as we go, what modes of ATP synthesis are being used? Eventually, now we actually, yeah. Now, the thing is, eventually, this is going to go, depending, again, it's all variable, it's all relative. We're going to end up going into this lactic acid production, okay, eventually. And that is a good place to stop. I think that's enough for now. All right, so the next video, there will be just one more video to cover this last part of chapter 11. All right, and I'll see you then. Have a great day. Bye-bye.